Good morning and good evening, everyone. I'm Takao Hensch. Normally, we would be welcoming you into our lecture hall in our building at Harvard, but unfortunately, we're living in strange times. However, there is a silver lining, and that is it makes it possible to have this conference call across the globe, and many of you are from all over the place, and so I'm welcoming you to our virtual event. This year's topic is one of great global significance, as always. In this case, we're discussing mental illness and the global burdens of mental illness from a new perspective. As many of you know, neuropsychiatric disorders contribute the most, perhaps, to disability-adjusted life years lost around the world. This is a slide from a few years ago summarizing North American situation, where you can see that neuropsychiatric disorders actually are more than the sum of cardiovascular diseases or cancers in terms of these dailies lost. And it's a very severe situation which is escalating around the world, most acutely now thanks to a new presence in the world of coronavirus. Mental illness is the leading cause of disability-adjusted life years around the world increasingly. And in the time of corona, with the social isolation, increased anxiety about the uncertain world, and of course the possibility that Covet invades the brain, we have a second epidemic coming shortly of mental health concerns. And so this topic is of great global significance. It has drawn attention for the past several years, thanks to advances in technology, spearheaded by the Decade of the Brain in the United States, for example, on two ends of analysis. At the molecular level, genome-wide association studies have linked a number of factors, mostly at synapses, to mental illnesses. And on the far right, technologies for non-invasive imaging and ever finer behavioral phenotyping have improved the characterization of the presentation of these mental illnesses. But as we all appreciate, there is an enormous gap between molecules and behaviors. And the goal of current neuroscience in this field is to fill this gap. We and many others focus on the microcircuits of the brain, and trying to understand mental illnesses as connectopathies or circuit miswirings or circuit malfunctions, including plasticity and dynamics of these circuits. And so increasingly, this topic is facing a challenge of complexity at multiple scales. Big data science has definitely arrived in the neuroscience field, and we need computational approaches in order to tackle this information overload, as well as to come up with new principles that are involved in brain development and brain disorder. So how do you tackle something so complex? One approach is to use animal models, of course, and that's the work of the Conti Center at Harvard, one of the sponsors of this event today. 4 PIs at the Conti Center, which is funded by the National Institute of Mental Health, are looking at the consequences of the same disease genes, how they play out in the development of animal models such as mice or marmosets, and in human tissues such as organoids in culture, or even pathological tissues from patients. And ultimately, what ties us together is a search for the circuit miswiring that may occur as a result of any of these risk genes that may play out either similarly or different in different species. But an overriding theme is the one of development. We think that mental illnesses have origins in early life, no matter how late they manifest themselves. And in fact, this sort of research gives us an opportunity to think even broader. In fact, our human behaviors as adults are sculpted during critical periods of early brain development, leading from even before birth through adolescence. And this is a time which is ripe with opportunity for understanding how neural circuits are normally wired up to produce the behaviors that we consider neurotypical. This is also an opportunity to learn principles for next generation artificial intelligences, which might actually leverage some of these ideas and create the possibility for super intelligent behavior, for example, which might catapult our human evolution and human studies. But in fact, we deeply need to understand these developmental principles in order to understand when trajectories of development go wrong. And once again, the ability to understand this at a deep computational level may someday allow us to correct impaired trajectories of development. And so today you'll get a flavor in the talks of how we can start to promote resilience against disorders and disease and predict in advance when such conditions are presented in people who are at risk. Another opportunity, as I said, is to promote therapy once symptoms emerge, and perhaps one day in the future we'll be able to use these insights to enhance cognition. This work is carried out in partnership with the International Research Center for Neurointelligence based at the University of Tokyo. And this is one of about a dozen world premier international research centers supported by the JSPS. A key feature of these WPI centers is its international global outreach. And this is an example of our research network, including Boston Children's Hospital and the Harvard Reischauer Institute. A key feature of our science and these networks is the exchange of scientists. And over the past few years, we've been able to send students to Japan, as well as receive medical students from Tokyo to Harvard and build this dialogue extensively. Sadly, the coronavirus is putting a damper on this, but we hope that events such as this one will continue to stimulate all of you to consider these kinds of exchanges as you develop your own careers. Finally, I will thank our two speakers today who very graciously accepted to speak very early on Sunday morning in Japan, but thanks to the Zoom, are able to join us at this event today. You have the bios of both speakers today. I will just mention very briefly that Dr. Kiyoto Kasai is a professor of psychiatry at the University of Tokyo and member of the IRCN project. And Dr. Mitsuo Kawato is the director of the ATR in Kyoto. And he is well known for computational approaches to understanding the brain. So we thank you again for joining us, Drs. Kasai and Kawato today. And we look forward to hearing about translational science in the age of AI.
Thank you, Hensch Sensei. Next speaker is Professor Kiyoto Kasai from the University of Tokyo and WPI-IRCN. The title of talk is Neuroimaging Investigation
of the patient. And schizophrenia is characterized by positive symptoms and negative symptoms and cognitive impairment. And the onset of the disease usually occur at around adolescents and young adults. And schizophrenia largely affects young people's life. The health status with the highest disability rates were acute schizophrenia, and the total cost of schizophrenia is huge in Japan and also worldwide. And biochemical circuit in schizophrenia is associated with the hyperdopaminergic in subcortical structures, and it may be related to positive symptoms, and hypodopaminergic in prefrontal cortex, which is corresponding to negative symptoms, one for schizophrenia. And it explains the onset of schizophrenia as the genetic plus early environmental factors, including winter birth and virus infection, low birth weight and hypoxia at the perinatal period. And then they go through the normal development, and they experience the first episode of psychosis in adolescents and young adults. However, from late 19th and early 2000, the neuroimaging technology have been so developed that we have been able to assess the precise gray matter volume reduction in schizophrenia and other psychiatric disorders in vivo. And we could measure these kind of biological indices through the clinical stages. And among such studies, we have been done the structural MRI imaging in a patient who experiences the first episode of psychosis. And this slide shows the my earlier studies almost 20 years ago, actually in Boston, and under the supervision of Professor Marty Shenton at Harvard, Brigham, and McLean. And what we did was the precise measurement of gray matter volume of the superior temporal gyrus. I use a laser pointer here, and superior temporal gyrus, which is associated with auditory hallucinations. And we scanned the MRI at 2 times. And also we assess EEG. And what we found was the schizophrenia was associated with progressive gray matter volume reduction over time through 1.5 years in the first episode psychosis period in auditory cortical gray matter volume as compared with affective disorder patients. And we do not still know the precise underlying mechanisms for the gray matter volume reduction in psychiatric disorders, but there's some indirect evidence that the post-mortem studies found that synaptic dendritic spine reduction in post-mortem brains of schizophrenia in prefrontal cortex and also in auditory cortex. So we further studied the functional auditory cortical dysfunction in schizophrenia by using the paradigm of mismatch negativity. I will briefly describe the paradigm for that. And the participants hear the listen to the sounds sequences, and sometimes there are deviant stimuli, and for participants it is unpredicted. So the auditory cortex and prefrontal cortex activated by this type of unpredicted deviant stimuli, and which the mismatch negativity is realized in the auditory cortex and also in prefrontal cortex. And mismatch negativity is a mixture of adaptation to standard predicted stimuli and unpredicted deviance detection stimuli. So we have to delineate the 2 components. And recently we have found that schizophrenia is associated with impaired deviance detection component, which is the main cause of mismatch negativity, event-related potential reduction in schizophrenia, which has been found many times in other studies. And mismatch negativity is a useful indices because it is associated with glutamatergic neurotransmission. So in psychiatric disorder studies, we use mismatch negativity as an index of glutamatergic neurotransmission. And we did mismatch negativity recording in first episode psychosis patient and found that first episode schizophrenia patients were associated with a progressive reduction of mismatch negativity amplitude over time through the first episode of psychosis. And also we have found a correlation between gray matter volume mismatch negativity amplitude reduction and supertemporalized gray matter volume reduction over time only in first episode psychosis patients. And this type of first episode study have stimulated the investigations worldwide for the studies for at-risk mental state. It is attenuated psychotic status and thought of as a prodromal stage before the onset of psychosis. And we recorded again mismatch negativity in this critical period for developing psychosis, and mismatch negativity amplitude was already reduced at this stage. And also plasma glutamate level was elevated at already at-risk mental state period. And there was an association between elevated plasma level of glutamate and mismatch negativity amplitude. And another index we frequently use in schizophrenia research is 40 Hertz gamma band oscillation called auditory steady state response. And we also recorded this index in at-risk mental state patients and the first episode psychosis patient, and we found a significant decrease in gamma band oscillation power in early stages of psychosis. And this 40 Hertz gamma oscillation is thought as an index of a GABAergic neurotransmission deficits in this disorder. And mismatch negativity and 40 Hertz auditory steady state response are used as representative biomarkers for schizophrenia recently. So our previous studies of neurophysiological and neuroimaging studies on early stages for developing psychosis contribute to the modification of neurodevelopmental hypothesis of schizophrenia. And recently identified risk factors and brain pathology specific to each clinical stage for psychosis has been increasingly important in schizophrenia research. And this summarizes the what we found in human studies. We found a mismatch negativity amplitude reduction, which may be related to glutamatergic neurotransmission dysfunction, and MRI gray matter volume loss in auditory cortex may be related to synaptic reduction. And ASSR power reduction may be associated with GABAergic interneuron dysfunction, but these human and animal evidence are always indirect, and there's no direct evidence for that. So we have to have a so-called translatable neuroimaging and neurophysiological markers that could be measured using common techniques in animals and humans. And I'd like to introduce some of the results from these kind of studies. In Japan, AMED have studied a project focusing on non-human primates called common marmosets, because this animal have a large prefrontal cortex, and so and we measured mismatch negativity using human ECG. These patients are pre-surgical epilepsy patients, and we found mismatch negativity at around the superior temporal gyrus. And we have collected multi-center MRI and used this MRI sample. And we have been also collaborating with Dr. Mitsuo Kawato's group, especially in terms of computational approach. But here we briefly introduce our meta-analysis of subcortical volumes in schizophrenia using a very large sample size. And what we found was the unpredicted finding of globus pallidum, one subcortical structure volume increase in schizophrenia patients. And we tried to replicate the finding through the clinical stages of schizophrenia, and we could replicate this finding at risk mental state patients. And also, we have replicated this finding in first episode patients. And our next question is this might be replicated in adolescents, normal adolescents with subthreshold psychotic symptoms. So we conducted adolescent cohort studies in Tokyo, and this is called a Tokyo Teen Cohort, which is a random sampling of 3,000 10-year boys and girls from a general population in Setagayaku, Mitakashi, and Chofushi. And for subsamples, we have collected 300 MRI scanning and a longitudinally follow-up every 2 years. And what we found was the left globus pallidum volume increase in adolescents who have subthreshold psychotic symptoms compared with those who do not have this kind of symptoms. So it was unpredicted, but we have nicely replicated the left volume globus pallidum volume increase in adolescents and at-risk mental state, and the first episode of psychosis and chronic stages of schizophrenia. So we are now thinking about a new hypothesis for development of schizophrenia, and this is accumulation of dopamine D2 receptor dysfunction by social stress overload in adolescence may lead to a development of psychosis. And this hypothesis warrants further investigations with collaboration with basic neuroscientists, and we are now doing mice studies. So this is a conclusion slide, and neurophysiological and neuroimaging studies have shown glutamatergic, GABAergic abnormalities in the neocortex in early psychosis, which suggests altered EI balance in adolescence, the second critical period of human brain development. And globus pallidum volume increase may be a new promising biomarker that reflects aberrant dopaminergic circuits and warrants further basic studies using animal models. These neurophysiological and neuroimaging markers are applicable to both humans, animals, namely translatable brain markers, will lead to deeper understanding of pathophysiology and developing effective early intervention for psychosis. This is the acknowledgement. we owe many domestic and international researchers in terms of neurophysiology, neuroimaging, and Marmoset project and Tokyo Teen cohort. Thank you very much for attention.
Thank you, Kasai sensei. Last speaker is Professor Mitsuo Kawato. He is the director of ATR, Brain Information Communication Research Laboratory Group. The title of his talk is How Brains Can Learn from a Small Sample and its Application to Computational Psychiatry. Kawato sensei, please.
So I would like to express my sincere thanks to the organizers, especially Takao-san, for inviting me to this exciting Japan-US Science Forum in Boston. I'm still in Kyoto. So today's title is How Can Brains Learn from a Small Sample and its Application to Computational Psychiatry. So today's topics is current AI faces cost of dimensionality, and humans can learn from a small sample. And we developed generalizable brain networks biomarkers of mental disorders. And finally, I talk about decoded neurofeedback for causal neuroscience and precision medicine. The first topic is current AI faces cost of dimensionality. So most of you are well familiar with the success, but very small number of people are familiar with failure of artificial intelligence. AI is a combination of machine learning, big data, and fast computing. And the most famous example is this Google AlphaGo developed by DeepMind, which is a combination of deep neural networks and reinforcement learning called Q-learning, so DeepQ. And it beat human champions quite badly in the past several years. So AI is great with a huge amount of available training data. In this case, simulated tens of millions of games of Go. But AI is still quite miserable in robotics. In the case of humanoid robot with many degrees of freedom, state space is huge. The learning system should be huge too, meaning that it contains a huge number of plastic synapses. But in this case of robot demonstration, 1,000 failures are unrealistic. If robot falls down like this, researchers need to repair it with a huge cost and time. So this is an NG video of DARPA robotic challenge. The robot need to fulfill 8 different tasks, simulating disaster after Fukushima nuclear power plant destruction. As you can see, many humanoid robots fail, and a small number of humanoids accomplished all tasks very slowly, almost 10 times slower than humans. And main reason of this is so-called overfit or poor generalization. So if you have data, this is from MIT, by the way. If you have a data taken from second-order polynomial, and if you fit it with a linear equation that is first-order polynomial, you have a systematic error. If you use second-order polynomial, you have nice fit. If you fit it with, for example, 6th-order polynomial, all the curve passes through the data points, but you have wide oscillation in the area where data is not available. So this is generally observed when you increase the model complexity, that is a number of parameters or number of synaptic plus synapses, then the error for the training data monotonically decreases. But the error for the test data, which is called generalization error, first decreases, but then increase like this. So this is a poor generalization or overfit. And usually generalization error in a simple system is described as the degrees of freedom of the learning system divided by twice of the number of learning system. So in the case of deep learning, usually, if you have n, like 10 millions of images and texts and simulated games of Go, D could be quite large. But in the case of robotics demonstration, N could be not that large, then you have all these difficulties. If you prepare hierarchical reinforcement learning, just like my colleagues Jun Morimoto and Kenji Doya have already done in 2001 like this, then within 100 actual trials, the robot can stand up. But in this case, researchers needed to carefully prepare the hierarchical structure and what is the sub-goals and what is upper level goals and so on, which is not affordable in the human brain. So next, I'd like to say that humans can learn from a small sample, even for a gigantic problem. Human motor learning from a small sample, really, and how? It was demonstrated that human babies fail only 3,000 times in a lifetime. But people say that this is mainly genetically wired because horse babies can stand up just after the birth. So this is my grandson in September. At that time, he was still 9 months old and couldn't stand by himself, couldn't walk by himself. But according to his mother, my daughter, within 10 trials of failures, he learned this entirely new behavior to step down from the couch to the floor. But still, people could argue that some parts are genetically wired. So I was sure that within 200 trials, humans can learn entirely novel and very huge reinforcement learning problem. And AI people noticed that, like brain, we need modular decomposition hierarchy, and probably finding the light and low dimensional state space is the most important. Several people, including us, say that metacognition, abstraction, and consciousness are the key for this capability of learning from a small sample. So our basic questions was human learning from a small sample. Is it real? And how brains learn from a small sample? So what we did is experimental demonstration that humans can really learn a huge degrees of freedom reinforcement learning program from a small sample without genetic information. And as I said, statistical learning theory indicated that the generalization error is a learning system degrees of freedom D, or number of plastic snaps D divided by twice of a number of training sample. So for huge degrees of freedom program, still, if we require only a small number of training sample, reducing D is the only way to circumvent the curse of dimensionality. So we will show that metacognition in those lateral prefrontal cortex and reinforcement learning in basal ganglia somehow synchronize in information for learning to reduce D. So we picked up a very artificial and not genetically encoded program. That is, we pick up 200 box cells from arbitrary part of the brain and make an arbitrary decision boundary utilizing a machine learning technique. And one brain state was classified either into class 1 or class 2. And question was, can the brain find low dimensional reinforcement learning state space, which was arbitrarily selected by us and entirely unknown to our human participants. So this is online decoder-based reinforcement learning for 3 days. This is related to decoded neurofeedback, which I'll talk 10 minutes later. And this is decoded neurofeedback like reinforcement learning. So we defined either those lateral prefrontal cortex or visual cortices as a target brain area. And from this area, utilizing a matchbox cell decoder, we can determine whether brain state is encoding the rightward random dot motion or leftward random dot motion. And on all 3 days, day 1, day 2, day 3, this right or left information determines the 2 reinforcement learning state, which is entirely hidden to our participants. For leftward movement brain state. Option B, action B is optimal. But for the rightward movement state, action A is optimal. And only on day 3, this brain state determines also the physical movement of the random dot motion. But on day 1 and day 2, we presented only very dim, entirely random dot motion. So day 1, day 2, there is no physical clue about the brain state at all. And subject task is like this within a single trial. We have a 6-second intertrial interval. And during this time, actually brain activity was measured by fMRI. And then we showed these dim random dot motion or clear random dot motion on day 3. And only on day 3 and later within this stimulus, it carries movement direction information. The subject task is to answer whether the random motion is rightward or leftward. But this was an impossible task because on day 1, day 2, there's no motion at all. And then they needed to answer their confidence level. 1 is least confident and 4 is the most confident. Then they needed to select the action or option A or B. And with the probability of 80% for the optimal action, they could earn 30 yen, about 30 cents per each trial. Within 3 days, they carried out about 300 trials like this. So this is reinforcement learning task. But the state is hidden to our participants. So to our big surprise, already on the second day where there's no physical information, they could learn this reinforcement learning above chance level. And furthermore, we found out that the correlation between the confidence and the matchbox pattern analysis distance of the brain state from the decision boundary of direction decoder is statistically significant already on day 2, meaning that somehow their confidence reveals the distinctiveness of the brain state for reinforcement learning. And also high and low confidence in those lateral prefrontal cortex matchbox patterns are synchronized with small and large reward prediction error in basal ganglia matchbox patterns respectively, meaning that if you have high confidence in those lateral prefrontal cortex, it corresponds to the low reward prediction error in the basal ganglia. So reward prediction error is like a teaching signal or error signal in reinforcement learning. Low reward prediction error, meaning that reinforcement learning has already attained a lot. So somehow those lateral prefrontal cortex knows that if it is confident about the perceptual decision making, reinforcement learning has already attained a lot, and vice versa. And this tendency was not observed at all on day 1, but on day 2 already we have this clear separation. On day 3, it was even exaggerated. So can brain extract low-dimensional reinforcement learning state from an almost infinite space and learn huge reinforcement learning problem from a small sample? The answer is yes. And is metacognition related and supportive of reinforcement learning? The answer is yes. And so dimension reduction on feature selection was supported by brain-wide search and converging in prefrontal cortex and basal ganglia loop synchronization. That's what we found. So we can say that humans can really learn from a small sample, a very big problem, which is not genetically encoded. So the third topic is generalizable brain network biomarkers of mental disorders, which is very much related to Professor Kasai's talk. And we have unfortunately unmet patients' needs in psychiatry. So diagnoses such as DSM-5 are categorical and based on symptoms and signs without biological examinations. So high rates of comorbidity are observed and weak correspondence between categories and medications. No mega blockbuster was developed in the past 30 years after SSRI, serotonin selective serotonin reuptake inhibitor. That was a big success. And majority of patients are not fully cured lifelong, unfortunately. And so NIMH in the United States started research domain criteria, so-called RDoC in 2010, which was led by the former NIMH director. And the objective of this RDoC was biotyping based on genetics and neuroscience. There were several very important publications from this RDoC, but unfortunately, none of them was reproduced in independent cohort. In Japan, we had two periods of SRPBS, 2008, 2013, 2013, and 2018. And now we are in Brain and Minds Beyond from 2018. So we have been working for a similar issue for 12 years. So in research domain criteria, people believe that in traditional diagnosis of mental disorders, we have actually many different biological types of subtypes mixed for each disorder category. So with data-driven classification, we could find clusters which are biologically coherent. We have some very important publications, but none of them was reproduced in independent validation cohort. So in human neuroscience, resting state functional connectivity was one of the very big achievements in the past 10, 20 years. So we ask our subject to stay calm in fMRI, and functional connectivity was defined as a correlation between two brain lesions. If the two brain lesions synchronize, have a high positive correlation, we say these two areas are positively connected. If they are not correlated, there's no functional connectivity. If they are anti-synchronized, negatively synchronized, then we say functional connectivity is negative. There are many ways to partition the brain into many areas, anatomically or functionally. For example, if you use BSA and AAL to partition brain into 140, then you have about 10,000 functional connectivities. So we use these functional connectivities as a base for brain circuit biomarker for mental disorders, including developmental disorders. But we have difficulty of AI in psychiatry. That is, we observed huge site differences in MRI. And if you use AI to a single site small data set, you often observe overfitting and poor generalization. And clustering is more difficult. Actually, clustering is the objective of RDoC, but it is more difficult than classification. So until recently, neither patient classification or stratification was generalizable for perfectly independent cohort. No reproducibility in science equals to no practical utility in diagnosis or interventions in everyday medical treatments. We found out that harmonization and multi-site database are essential, and they can reduce this huge site difference as well as difficulty of AI in psychiatry. So in SRPBS, we developed a so-called DECNEF database project, and 2,409 March disorder participants were collected from many different sites, including Professor Kasai's University of Tokyo. And we also obtained 411 traveling subject samples. And this data set was made publicly available. And in collaboration with Dr. Kasai, we developed fast ASD biomarker. So from 10,000 functional connectivity, the AI can automatically select only 16 functional connections. And the linear weighted summation of this 16 connections determines likelihood of ASD. So for a typically developed participant, we have this kind of white distribution. And for ASD, we have this black distribution. Setting up the threshold at 0, we can classify TD and ASD with 80%, more than 80% correct. And the interesting thing is that this biomarker was developed utilizing only Japanese data set. But without any change of this biomarker, it can classify TD and ASD from 6 US sites with a percentage correct of about 70%. So this is a generalizable biomarker of ASD across the Pacific Ocean. We also developed brain circuit biomarker for melancholic type of depression. And the most exciting finding is that it picked up two very important functional connectivity. One is left lateral prefrontal cortex and left precortex. And the second one is right dorsal frontal area and left lateral and ventral frontal area. So for the first functional connectivity, healthy people have negative correlation, but melancholic type major depressive disorder patient has positive value. And after 6 to 8 weeks of application of SSRI, this functional activity got worse. So it was even larger than this initial state. After euthymic state, it returned to the normal negative one. For the functional connectivity 2, already at 6 to 8 weeks, this functional connectivity gets closer, so better to the normal. So we know that SSRI is efficient for some patients, but not for some patients. And around 6 to 8 weeks, some patients get worse and even, plan suicide. So now we are working on this functional connectivity 1 with sophisticated real-time fMRI neurofeedback therapy, which is related to the next topic. So the final part is decoded neurofeedback for causal neuroscience and precision medicine. So Takao Watanabe and I started this decoded fMRI real-time neurofeedback. Our question was not related to psychiatry at all 10 years ago. So our basic neuroscience question is, are primary and secondary visual cortices plastic enough to accommodate visual perceptual learning? Because there was long-standing controversy, where is the locus of visual perceptual learning? So we utilized this decoded neurofeedback technique to induce some merge cell neural activity in V1 and V2 corresponding to the specific orientation of gratings. As a result of this, we can induce perceptual learning. So this is not conventional correlation study of fMRI. This is a causal study. We change the brain activity. As a result of this, we can induce perceptual learning. So the trial's design of DECNEP and functional activity neurofeedback is like this. This shows a timeline of a single trial. So decoding of or functional connectivity computation of fMRI measured brain activity is the key of this technique. So we have intertrial interval of 6 seconds and fMRI measurement from 6 to 40 seconds, and this is called induction period of DECNEP. And then we compute decoding of functional connectivity, and we have next feedback period. It was terminal monetary reward. So after decoding the brain activity, likelihood of the targeted brain dynamics is computed. And the size of this green disk is proportional to the reward, actually monetary reward. And within a day, we have 50 to 200 trials. and several days, there's no instruction to a participant, and they were not conscious about induced information. With this coherent technique, we first, as I explained, induce visual perceptual learning and association of color and orientation, fear, memory extinction, while we can increase or decrease metacognition confidence in prefrontal cortex and parietal cortex. We can increase or decrease facial preference by manipulation of the cingulate cortex. And we even did intervention for animal phobia in double-blinded randomized controlled trial paradigm. Now it is extended to PTSD therapy. The important thing is that there's little information leak of facial preference from cingulate cortex to other regions in the induction period, while it is widespread in the decoding construction session. So we hypothesize that the consciousness requires information spread from the target area to probably prefrontal cortex. So human causal system neuroscience with DECNEF was established. So DECNEF is versatile and efficient in manipulating different cognitive functions of various brain regions with good effect sizes. DECNEF is a neural operant conditioning combined with decoding technique, and fMRI matchbox pattern, principal component analysis, and link architecture network simulation suggests that spontaneous and target neural activity is reinforced. So DECNEF is better than optogenetics for several aspects, including human usage, spatial temporal control, derived from human hundreds of brain activity patterns, and information specified by decoding. So this is a summary slide. I explained that current AI cannot learn large-scale problems from a small training sample. In contrast, humans can do above with metacognition. And psychiatry did not make a great progress last 30 years and needs help from neuroscience and AI, I believe. And we developed generalizable brain network biomarkers of mental disorders, and which were built through harmonization and sparse machine learning. And decoded neurofeedback for causal human neuroscience can be also used as a tool for data-driven and precision medicine intervention. Thank you so much for your kind attention.
Thank you, Kawato-sensei. Before we move on to the questions and discussion time, we'll take 1 minute. Please use this time to review the questions and submit the questions.
So I want to share a couple of questions we received. So first to Kasai sensei. The question is, do you think what kind of factors can affect adolescent brain development seriously? Food, human relationship, or education?
Yes, this is a very good point. And this is what we should know for further studies. But we believe that social interaction, like peer relationships, is very important for adolescent development. In child development, parent child bonding is very important for development. But for after the 10 years old or so, there's a big change in brain maturation, like prefrontal cortex, that make the brain sensitive to the what others think of ourselves. So social relationship is very important for adolescent brain development.
Thank you for answering the question. Next question is to Hensch sensei. How do you think this COVID era will affect our brains in very long time period?
It's of course a very good question, and we don't know yet since we've been living with COVID only a few months. There are two kinds of problems. One is the potential for neural invasion by COVID itself. I think it's becoming clear that this is possible. Certainly everyone knows about the loss of smell as a characteristic symptom, but postmortem brain tissue suggests that it does invade the brain. And this, of course, has direct pathological consequences. But the second is our response to COVID. We have, in many countries, gone into social isolation, social distancing. That is actually a risk factor for schizophrenia for people who are at risk. If this kind of situation continues, you can imagine people who are at risk to begin with will flip over into frank disorder or illness. Certainly everyone has anxiety, symptoms of fatigue, insomnia, and these things are all related, but are maybe secondary to our response. And so we need to get out of this COVID period as quickly as possible so that we don't exacerbate the risks.
Thank you for sharing your thoughts. The next question, it's kind of a comment to Kawato sensei. One of the participants comment, babies spend about a year between their birth and the first walk. So they do a lot of motor training beforehand. So that's not a small size sample. How do you respond?
So people could argue that learning from some movement can be extended to other types of movement. But actually, if you try to make humanoid robots to learn some task and generalize to other tasks, this is extremely difficult. So from so-called technical terminology, we need to build good internal models and to explore the state space for new motor skills. So if you are not practicing locomotion, you are just crawling on the floor, there's no good way of generalization from crawling to biped locomotion. So robotics AI did not succeed in these good generalization or learning of multiple tasks coherently. So we still do not understand how humans or animals have this capability. Thank you so much for a good comment.
Thank you for answering the comment. The next question is to Kasai sensei. Does the asymmetric auditory functions in brain exist in other animals or only in human?
Yes, there are a few studies examining the brain asymmetry in monkeys, but left lateralized auditory cortex is much more evident in humans. So there's less left-right asymmetry in other species than human beings.
Thank you for answering the question. One more question to Kasai sensei.
Yes.
Is it possible to stimulate specific regions in the brain so that we can improve specific functions?
Yes and no. So we cannot do the invasive stimulation to the specific brain regions. For example, repetitive transcranial magnetic stimulation called a TMS can stimulate a prefrontal cortex, but it's not so specific. So we do need more precise stimulation or modulation, which is the one of the promising tool is decoded neurofeedback that Dr. Kawato nicely presented in this talk.
Thank you for answering the question. So this would be a final question to Kawato sensei. I guess the question is, I guess some animals have less neurons, but still behave without many error. How do you think about it?
Yeah, actually, the number of neurons is quite tricky. So if you have huge number of neurons, it means that you have large degrees of freedom in the learning system. And as I said, if you have large degrees of freedom in the learning system, you need huge amount of training data. So I think if the lifetime of some animals is short and do not need to explore all different possibilities of learning, small number of neurons is beneficial rather than harmful. So I think, humans have large degrees of freedom, and so we need to do many different things fluently. Thank you so much for this interesting question.
Thank you for answering the question. So thank you very much for all the presenters and the audiences sharing your questions. So we cannot actually meet each other, but please give your hand, react to the presenters.